Hi, my name is Emily Neal, and we are talking about how to interpret your pyrosequencing data in this video from Lab 10. And this is intended as a follow-up video to help you analyze your data. But for writing up the report, we would ask that you also, or suggest that you also watch the intro video again on microbial source tracking. Getting back to interpreting your data, remember the expectation is that pyroprints within each group should be exactly the same, because each member within a group had analyzed E. coli from the same exact isolate. So we're going to use and analyze these pyroprints to help us answer the question, how reproducible is pyroprinting for a particular E. coli strain? So we're going to compare pyroprints within the group, again, to answer this question. So let's take our hypothetical group number 1 here. And the first thing I want you to know is to not pay attention to the sequence that's in the upper left-hand corner on each pyroprint. And just to orient you on these pyroprints, each one of these histograms is a separate pyroprint. So the first individual in a group, human 190A, that's the pyroprint for their particular isolate that they analyzed. Okay, so we're going to compare these two different panels. Remember, within a group you have 3 people, so you're going to be comparing this across all 3 of your pyroprints. But again, I don't want you to pay attention to the nucleotides up there in that left-hand corner. Just pay attention really to the histogram itself. To not worry about the differences in the color. So blue versus yellow versus red, those colors don't really mean anything for our particular application, so don't worry about where those colors begin and end. For example, these 2 bars that I've highlighted here, these are basically exactly the same, but they just have different colors. So we would not consider that any different. What we're really looking for is peak heights. We're going to compare the peak heights. And so we're looking for large differences of the peak heights, and that's what we would consider something different between the pyroprints. At dispensation 48 here, this is a T. This is just below, or the machine has called out about 1 nucleotide, and if you look at the corresponding peak in the next pyroprint, it's roughly about the same place. So again, the machine would have called out 1 nucleotide, or 1 T, for that particular location. The differences between them are very subtle, so this is not something that we would consider a difference. And if you went through the entire pyroprint, you would find that there are really no major differences between them. So these pyroprints are would be considered a match. If we looked at our next group, group number 2 here, again we have 2 pyroprints from the exact same isolate, human 147. And if we start looking for differences, let's look here at dispensation 16. We dispensed a T at this particular dispensation, and the machine called out just above 4 nucleotides, 4 Ts for this particular location. But if we look at the corresponding peak at that location, we've called out about 4.5 Ts for that location. So there is a difference there, although it's not a huge difference, so I'm going to put a question mark there because I'm not sure how much of an impact that is really making on the reproducibility of this particular method. The next difference is a little bit more obvious though. At dispensation 36 here, we dispensed another T there. And if we look at that same dispensation on the other pyroprint, you'll notice that there is no peak whatsoever. So in the second pyroprint in 147C here, the machine called out 1 nucleotide, 1 T there, whereas on 147B it called 0. There was no T identified by the machine. If we look at dispensation 40, we'll notice that the machine called 4 Ts in this particular location, but in the next sample, in 147C, the machine actually called less than 3 nucleotides. And so that's something we would consider a major difference in the height of the peaks. Dispensation 49 here, there's a C that has been dispensed at this location, and the machine is calling that about 1.5 Cs were added to that extending strand. But if we look at that same position in the next pyroprint, you'll notice that there is a gray bar there. We consider gray bars to be 0. The machine basically doesn't recognize them, and so it noticed that, or it called out 0 Cs being added. So again, this is something we would call a major difference, the presence or absence of bars. If we continue down the line here, we'll notice that there are continued to be differences in the number of nucleotides the machine recognized as being added to the extending strand. And so if we progress outwards here, more and more of those differences are appearing until we finish out the entire dispensation order. And as you'll notice, there are quite a few more differences between these two pyroprints than there were between the first two pyroprints from group number 1. So how do we interpret that? What do those differences mean? Remember, again, we expected that pyroprints were to be the same within the group because the E. coli was from the same isolate. Well, for group number 1, remember that those pyroprints had no major differences, so we consider those pyroprints to be a match. And we know that human 190A and human 190B are the same. They were genetically identical E. coli. Therefore, we would conclude that reproducibility of pyroprinting appears to be high for that particular group. Now, group number 2, if you had data that looked like that, you would say something along the lines that pyroprints have greater than 5 differences, so we would not consider them to be a match. And we already knew that human 147B and human 147C were the same. Again, they were from the same exact isolate, so they were supposed to be genetically identical. So we would conclude then that the reproducibility of pyroprinting appears to be low for that particular group. These are the things that I look for when I'm analyzing the pyroprints and looking for differences. Don't be afraid to draw on your pyroprints, circle, arrow, question marks, notes to yourself, whatever you need to clearly identify those differences. And as always, don't be afraid to ask your instructors for help if you have any questions.